right, good afternoon. It's 3:10, so we're going to get started. I'm Dr. Kay Stefanik, Assistant Director of the Iowa Nutrient Research Center. I want to welcome you all to our spring seminar series, Insights from Next Generation Water Researchers. Each seminar this semester is going to have two speakers who are either current or recent graduate students or postdocs who have worked on INRC funded research. Before we begin, I just have a few housekeeping items. The first is this is a hybrid meeting, so there are people online. For those of you online, please type your questions into the chat box, and I will read them out to the presenters at the end of their talks. For those of you in the room, you can just feel free to raise your hand at the end of the talks and ask your questions directly. And then the second item, this webinar will be recorded and posted to INRC's website along with previous seminars. So with that, our first speaker is Grace Jackson. Grace is a master's student in Dr. Charlie Lowen's lab in the Department of Ecology, Evolution, and Organismal Biology here at Iowa State University. She's going to be presenting on restored wetlands. How does vegetation and nutrient reduction affect algae communities?
Thank you. So, what are these restored wetlands? Give me a second. It was on. Yeah, it does say that it's on. Okay, we're just going to use this. No worries at all. Okay, so these wetlands are through the Conservation Reserve Enhancement Program, and it's this huge partnership with Iowa Department of Agriculture and Land Stewardship, USDA Farm Agency, and other state, local, and federal entities. And this program incentivizes landowners to construct wetlands in order to capture agricultural runoff. In order to qualify for this program, your land needs to reside within the Des Moines Lobe. This is a region with characteristically poorly drained plains due to recent glaciation, and it's a perfect place to put a wetland. Here's an example of a CREP wetland. We have a stream capturing runoff from lots of farm field that flows down into this main basin and then continues on downstream. So my first goal is to investigate the effects of nutrient removal happening within the wetlands by comparing algae communities up and downstream of the main basin. And then my second goal is to characterize the communities within the basin itself in order to assess the ecological health and conservation value of these wetlands. So why do they exist in the first place? As you know, Iowa is an agricultural landscape, and we provide 11 to 52% of the long-term nitrate load that eventually makes its way down the Mississippi into the Gulf of Mexico. Algae blooms occur, which end up dying. They are decomposed, and it creates a very low oxygen environment that's bad for fisheries, among other wildlife. But thankfully, these CREP wetlands are very good at removing nitrates and other contaminants. Here's an example of how CREP is a sink for nitrates. So we have inflow nitrate loads on the dark dotted line and outflow on the open circles. And then this gray portion is the hydrology or discharge from the wetland. And as you can see, there's more nitrates coming in and less nitrates coming out. So how is this occurring? Well, there's an important part of the nitrogen cycle where nitrate is transformed into dinitrogen, a gaseous form, through microbial process of denitrification. This gaseous phase does not occur for phosphorus, another main nutrient that comes in from farmlands. Instead, phosphorus is decomposed, uptaken by the plants, or settled into the sediment. And this difference, we hypothesize, will result in different algae communities downstream after these processes occur within the main basin. So as I said, our first goal is to investigate the biological effects of this nutrient removal, and we're going to be using periphyton or attached algae to compare these communities and look at the complex responses to nitrogen and phosphorus. Why are we using algae? Well, this is an extremely important group that sustains the aquatic food web because it's at the base. So algae is grazed upon by zooplankton, fish eat zooplankton, so on and so forth. However, this important group can also become detrimental as it can become overgrown or contain harmful taxa such as certain species of cyanobacteria, which produce toxins. They are likely responding to these downstream changes in the key nutrients of nitrogen and phosphorus and can be used as indicators of change. This is how we're studying the periphyton. We have a temperature and light sensor in each stream, and we use this periphyton assay. These are microscope slides that are tucked into a flotation device, and they float on the water surface or just below it for about a month. We come back and we measure them using a handheld spectrofluorometer known as a benthotorch. This benthotorch understands, it fluoresces the slides, and it has a computer that knows the ratio of chlorophyll A and other pigments to identify three main groups of algae, which are green algae, diatoms, and cyanobacteria. So it's a really cool magic algae wand that we get to use. And we can use this information to study, is the wetland improving water quality? Algae is a part of this water quality. As I mentioned, some species can produce toxins or become nuisances, and we can learn a little something about ecosystem health. We have 9 of these CREP sites with 3 reference more natural wetlands, York-Boda Marsh, and a couple of wetlands in Union Hills. I took the concentration given to me by the benthotorch and divided it by the number of days that that microscope slide was deployed in the stream in order to get a growth rate of those different groups of algae. I calculated their standardized differences, which are effect sizes. And for your knowledge, early sampling period was in July and later was in August of 2024. Before we get into my results, let's go over the anatomy of a forest plot. So we have our wetlands on the y-axis, and then the effect size on the x. We have a point estimate of the difference between up and downstream communities indicated by the dot, and the line is our confidence interval. If that confidence interval crosses the 0, that means there is no difference in periphyton growth up or downstream. But if it's to the left, that means there was a higher growth rate of that taxa upstream. If it's to the right on the positive side, that means there's a higher growth rate upstream. So let's look at cyanobacterial growth rate. This is a group of algae that can sometimes become a nuisance. Just for your knowledge, if you're wondering why there's not 12 wetlands up there, sometimes we lost our samplers due to storms, so we did not include that. And if you see a point without confidence intervals, that is because a measurement was taken, but there was no growth of cyanobacteria up or downstream, therefore there was no difference. And these darker colors down here are the pooled estimates. So we averaged the effect size for season and then overall. So there's no trend in any of the pooled estimates. However, there are wetland-specific differences in cyanobacterial growth rate. All right, so let's look at wetland WW. Earlier in the summer, there was higher growth rate of cyanobacteria upstream, but this relationship shifted across the summer, and towards the end, there was higher growth downstream. Whereas in FCC2, there was just consistently more cyanobacteria downstream. In our reference site of Union Hills, there was never any difference. Now let's look at diatoms, another group of algae that are made of silica. We have a trend with higher growth rate of diatoms upstream across the season and an overall estimate. But again, those wetland-specific differences still exist, with some wetlands just not having any difference across the season of diatom growth up or downstream. Okay, and finally, let's look at our green algae. No trends in any of the pooled estimates, but I want to highlight that sometimes we can see opposite relationships depending on the wetland. So for SS, early in the summer, there was more green algae upstream, but this relationship disappeared later in the summer. Meanwhile, the flip side happened with LP, with no difference in growth early in the summer and then higher growth upstream later. Okay, so there are wetland-specific differences between up and downstream periphyton communities, but why? Well, there's a lot more to discover. I will be using the environmental data of temperature, light, nutrients, and flow data to pinpoint possible drivers of these patterns that we just saw, using multiple regression, essentially finding correlations between these environmental variables and these periphyton growth patterns. I'm particularly interested in how the concentration of nitrogen and phosphorus over time can correlate with this growth. In addition to our environmental data, we will be utilizing the Crumpton lab data, as they have continuous sensors up and downstream for flow and nutrients. So what scale is the algae responding to these nutrients? Let's say we sampled in late September. Does algae seem to be responding more to this spike of flow just a few weeks before? Or maybe this longer low flow period is impacting the growth more? Or maybe it's some kind of overall mean for wetland. Now let's move on to the second goal, characterizing communities within multiple habitat zones to assess the ecological health and conservation value of these wetlands. So we know these wetlands are made to capture nutrients, but what other benefits might they be providing? Well, wetlands contribute to cleaner drinking water. They can be used for recreation, fisheries, and they contribute to carbon cycling. So we need to understand what else is happening in this wetland beyond just, is it capturing nutrients? And we can do that by characterizing the habitats, the flora and fauna within them. So we have three main habitat zones in these wetlands. We have emergent vegetation, which breaks the surface of the water, and an example of that is cattails. We have submergent vegetation, that's below the surface, such as coontails, or we have areas where there's little to no vegetation, known as the open water zone. Here's us characterizing these zones. This is Caitlin, she's in the emergent zone here, and the submergent there. We got a distance at different points of the wetland to measure the size of the zones every time we went, and then we also measured the depth at which each zone ended. The cool thing about these wetlands, and the challenging thing as well, is that they're very dynamic and they change throughout the summer. So here, it's pretty much a lot of open water, but the submergent zone started to encroach upon the open water zone, typically in these wetlands. And it sort of crowded this out. So the zones changed across the season, which is important to pay attention to. In addition to characterizing the vegetation, we also looked at lots of different forms of life. So here's Daniel in the emergent zone collecting his bugs. He's collecting everything in that trash can, which is kind of amazing. This is Caitlin with her Van Dorn to capture zooplankton in the open water zone. It's basically this acrylic tube with plungers on the end that trap the zooplankton. And then there I am with the YSI and pH sensor that looks at some of the chemistry, and then we also took bottle samples for chemistry as well as phytoplankton, which are non-attached algae, but they're the algae that you see when you see a bloom, and it's pelagically in the water. And all of this is part of understanding the co-benefits beyond just nutrient reduction. So right now, I'm characterizing the phytoplankton community using a tool known as the FlowCam. This is an amazing instrument. I put a sample of water It goes down a tube. There's a microscope and a camera, and it gives me lots of images of my different taxa. Here's some from our samples from this summer. And it allows me to identify and sort them very rapidly compared to traditional microscopy. This is a really good skill to have, as many monitoring programs use this instrument. And I'm having a lot of fun. There's lots of other professional development happening in this project. We have gained lots of field skills because we were out there all day and learned many, many different ways of collecting multiple different forms of taxa. There's also a lot of problem solving involved. This is a pretty muddy zooplankton sample, especially compared to a lake. And we had to make sure that we got everything in here without losing her microscopic organisms. So there's a lot of like rinsing of the fingers into the bottle and like holding things very tightly so nothing fell out. But Kaylin did an awesome job with that. And we also worked together to make sure we were placed on time.
Right.
And we really relied on each other. And it's a great learning experience. I'm also learning a lot about study design, how to answer the questions I have about these wetlands. I'm doing new forms of statistical analysis, new to me. And I'm practicing my communication skills right now in front of you. And I've gained insights into my own career goals. I really enjoyed learning about the intersection between human need and wildlife. And I hope to continue studying more in that realm. Thank you to the Lowen Lab members and the Iowa Nutrient Research Center for funding this work, as well as Iowa State, EEOB, and the Finch Fund for Botanical Studies. Any questions?
We do have time for one or two questions now.
It may not be related to what you do, but the CREP, the ones that are like promoting wetlands, is it like a financial, like do they like provide cost, like payment or something to help cover the cost of not building these on the farm, the farms that are going on?
I think they do, yeah. I don't know like the details of how much, but yeah, I'm pretty sure that there's a financial incentive.
Oh.
Yeah.
You measured 3 different types of algae, if I know nothing about algae.
Mm-hmm.
Why did you pick those 3? Like, are they indicators of something, or they just, like, so?
Yeah, so those 3 main groups perform different functions. It's also like what our instruments were able to rapidly read, but there are lots of indicator species within those groups. And we also collected the periphyton itself, and we'll hopefully be looking at more like species level things rather than groups. But it is good to know the relative abundance of these groups because they are associated with different water quality parameters. So, yeah.
What do you do with the bugs?
That's his realm. That's Daniel's realm.
Like, what, you have a trash can of bugs? Like, what do you count them? Look at them? Okay.
Yeah, I count them. I'm also looking at plants, and they're 2 looking associations with bugs and plants in all the different plants.
What's the coolest bug you found?
But maybe the coolest one is I found a snail parasite that lives inside the snails, and then it goes up into the snail's eye stalks, and then it pulses, make colors that attracts birds, and then birds eat it, and then birds it out. And then they get eaten by snails again, and then it kind of creates this really cool life cycle.
You got a really cool visual
No,
question.
Yeah, it is really cool.
It's much cooler than really.
I think one of the nice things about Iowa Nutrient Research Center is we fund all kinds of different projects, everything from manure management to more ecological projects. And I think maybe it might help make a connection if you could describe the different water sources coming into the wetlands and what those water sources might be bringing into the wetlands.
Yeah, so we got a really good visual of this after rainy days. where we would be walking across the farm fields to some of the harder to reach wetlands. And you could literally see just like veins coming off of the landscape into these streams. So I'm pretty sure they're connected to other water bodies within that Des Moines lobe. But yeah, I don't know. Is that kind of what you're looking for? Yeah.
And a lot of them would be fed by tile drain.
Yes.
Yeah.
Yes, that is.
But it is interesting that you saw the runoff.
Yeah.
Yes.
So you measured 3 different vegetation zones.
Mhm.
That's an apple question. But what do I want my wetland to look like?
Hmm?
What do I want my wetland to look like? Do I want a vegetation zone, or do I want open water, or?
So that's something that we're looking at as a lab. We are using our measurements of the relative size of these zones, and our plan is to compare them to the biodiversity of these different taxa, especially Daniel's bugs, because he has like actual scans of like each species of plant. He's going to relate the surface area of those plants to, I'm trying to remember, there's a lot that's going into this, but basically we are looking at the vegetation and like the overall construction of the wetland and trying to compare it to what we're seeing with the biodiversity. But yeah, not the best answer, but
No, that's probably what it might
Yeah,
level.
yeah.
That's certainly a question that I think we hear, you hear a lot about that, open water, there's no
Mhm.
value. So understanding, the biodiversity of these different zones and what it means is extremely important. Yeah.
Yeah.
Awesome.
Right. We will hold any additional questions till the end, and we're going to move on to our second speaker.
Thank you.
Here, I get it.
All right. So our second speaker is Dr. Chris Morris. Dr. Morris is a postdoctoral research associate in the Department of Sociology and Criminal Justice at Iowa State University. And he's working with Dr. Jay Arbuckle. He will be presenting on the social science of conservation practice adoption.
All right, good afternoon, everyone. I appreciate those of you that braved the blizzard to be here in person, and to those of you that are online or listening to the recording, thank you so much for tuning in. Thank you to Grace for that awesome presentation. I love to hear what my colleagues in the natural sciences are doing. I'm going to be talking about things from a sociological perspective today, and hopefully some of you can get some value out of that. So I'm going to tell you a little bit about where I come from, and then I'm going to speak briefly about the social science of conservation adoption, the history and the trajectory of that. And I'm going to tell you about a couple of different projects that are funded by the INRC that I'm currently working on. So my background originally, my grandparents on both sides of my family were farmers and ranchers down in Texas. But they had to sell the property before I could inherit anything. So I wasn't going to be a farmer, but I grew up working on farms and ranches, and I decided that if I couldn't be a farmer myself, I wanted to work with farmers in some way. So I got my bachelor's degree in range science at Montana State University, and then came back to Texas and ended up working for the USDA Natural Resources Conservation Service as a rangeland management specialist. So in that capacity, I worked in a county field office. And so my job looked like meeting with farmers and ranchers out in the field, developing conservation plans, providing technical assistance, and administering financial contracts for incentives and things like that, incentives to do conservation practices. So I made a career out of that. I was in that capacity for 15 years. And then I realized two things. Number one, although I think the NRCS does a great job on what it does, there's always ways to improve policy. I think that's something that we can all agree on. And number two, I realized that I had a passion for teaching, and I wanted to use that experience with the NRCS to develop research to help improve conservation policy and to teach the next generation of conservationists. And so I heard about the Sustainable Agriculture and Rural Sociology programs at Iowa State University and the work of Dr. Jay Arbuckle. And so I came here to Iowa State. I earned my master's in 2020 and my PhD in 2023 and had so much fun. I stuck around as a postdoc, still here at Iowa State University in the Department of Sociology and Criminal Justice. So in general, my research looks at why farmers make the decisions that they do and how we can use policies, programs, education, and outreach to encourage and support farmers to grow our feed, food, fiber, and fuel more sustainably. And so I'm going to talk a little bit about why I'm doing that, kind of the history of that. So the social science of conservation adoption has been around for a really long time. Some of the initial studies in social science in agriculture were done in the 40s, actually here at Iowa State University. This is one of the first studies by Ryan and Gross that looked at the adoption of hybrid corn varieties by farmers. And so they looked at various factors that influence how farmers get information. So on the left here, you'll see where they were tracking the influence of salespeople, neighbors, and farm journals. And of course, later we would start incorporating things like extension agents and conservation organizations. And then on the right, you'll see there they started mapping how much information is put out into the communities over time and at what rates farmers began adopting these practices. So in the beginning, social science was really invested in understanding how farmers incorporate these new technologies over time. There we moved on to really promoting some of the this is associated with the industrial model of agriculture. So again, some work here from Iowa State by Beal et al., looking at attitudes regarding farmer adoption of using commercial fertilizer. That might seem a little weird to think about that, but there was a point when commercial fertilizer wasn't widely used by farmers, and we were trying to understand how to get farmers to start doing that. Here's also a study about the use of 2,4-D spray for field weed control, trying to get farmers to use herbicides and pesticides. And so back then, the language was really about adopters who were incorporating these practices onto their farms, it being characterized as innovative, socially engaged, the smarter farmers. And there was a characterization of those that didn't adopt these new technologies as older, maybe not as smart, and they were referred to in the literature as laggards. Now we can look back at that research and realize that that was kind of problematic for a few different reasons. But basically, all of this type of research can be was summed up by Everett Rogers, who talked about the diffusion of innovations. A lot of people have heard about that. This is where you get the language of early adopters, mid adopters, and laggards. Some people that aren't even involved in agriculture or social science will use those terms, and that's where that comes from. So in general, the social science in agriculture back in the mid-century was really looking at how do we get farmers to incorporate these new technologies? And by gosh, they did it. We asked farmers to adopt new varieties, new hybrid varieties of corn, and they did. We asked them to use a lot of commercial fertilizer, and they did. We asked them to use herbicides and pesticides to increase their yields, and they did a fantastic job. And when I say we, I mean the land-grant universities, extension, agricultural organizations, as well as policymakers. And policymakers said, grow fence row to fence row, and they did. So farmers did everything that we told them to. Beginning in the 60s and 70s, we started really realizing that there might be some environmental issues with some of these industrial type farming practices. And so here in Iowa, we have soil erosion and depletion that occurs, on most acres in Iowa. We're losing literally tons of soil per acre per year at an unsustainable rate. We're seeing water quality, some of the things that Grace alluded to in terms of water pollution, either from surface runoff off of agricultural fields or from tile drainage. And that's not just affecting Iowa, that's also affecting our neighbors downstream. And so we have all of this accumulation of these nutrients, phosphorus and nitrogen mostly, ending up in the Gulf of Mexico, creating the hypoxic dead zone there and causing issues there. But it's not all doom and gloom. Luckily, there are a host of conservation practices that farmers can incorporate to make their farming operations more sustainable. So things like cover crops, no-till, edge of field buffers, infield buffers, wetlands, bioreactors, and saturated buffers. Unfortunately, farmers are not adopting these types of practices as quickly as they adopted hybrid corn. And so this data is a little bit dated. It's from a few years ago, but things haven't changed dramatically. So what this says, this is information from the Iowa Nutrient Reduction Strategy. It says, if we continue at the current rate of cover crop adoption, we will not meet our goals for nutrient reduction for 85 years. When we look at the amount of wetlands that you just talked about, if farmers adopt at the current rate, we're not going to meet our goal for 942 years. And if we're looking at bioreactors and saturated buffers, it'll take 22,000 years to meet our goal. So we need to push the needle forward, and that's where social science comes in. So why haven't we met these goals? Well, the fact of the matter is humans are really complicated. Human behavior is incredibly complicated. And as Isaac Asimov said, the human brain then is the most complicated organization of matter that we know. This is where folks like me come in, and not just me. This is a smattering of social science research that has been done, largely starting in the 80s and continued over the next 40 years, trying to figure out, like we did in the 40s, why are farmers making the decisions that they do, and how can we incentivize them to adopt these types of conservation practices that will help address these environmental issues? A couple of papers that I will point you to in particular are really good reviews of the literature. So Prokopi et al. 2019 looked at all of the quantitative data on farmer conservation adoption from 82 to 2017. So quantitative is looking at things like survey research mainly. And that paper kind of gives a summary of what we know, putting all of the literature together. What is our best knowledge with all of these different studies? And then I'd also like to point you to Ranjan et al. 2019, which did the same thing, but for qualitative research. And so when we say qualitative social research, we're talking about things like focus groups and interviews and things like that. So both of these are really great publications to kind of give you the state of the state regarding what we know. That being said, there's an upside and a downside. The downside is we don't know everything. There's still a lot of unanswered questions. The upside is people like me have job security. And that brings me to a couple of projects I'd like to talk to you about that were funded by the INRC. These projects have been a huge part of me getting my feet on the ground as a postdoc, and I'm incredibly grateful to the INRC for funding these projects. So the first study is a survey project. The title is called The Flux of Agricultural Conservation, Understanding Changes in Iowa Farmers' Adoption of Cover Crops and No-Till over Time. Obviously, cover crops and no-till having positive effects on water quality and nutrient reduction. And these are my co-authors. This paper is currently in a revise and resubmit. And our main research questions were, how does conservation practice adoption behavior change over time among Iowa farmers? This is something, believe it or not, that hasn't been looked at, largely ignored in the social science research on conservation adoption. We tend to look at just one point in time. Does the farmer currently adopt conservation practices or do they not? And then we draw conclusions from there. What we haven't looked as much at is what do they do the next year and the next year? The second question that we asked is, what factors affect these changes in adoption behavior? I'm going to be giving you some results from question number 1. Question number 2, we are crunching the data as we speak, and that will be the next paper. I'd be happy to come back and talk to you about that once we get that data as well. So this project, we did surveys in 6 HUC6 watersheds across the state of Iowa over a period of 5 years. Not every farmer was surveyed in every single year. And then the other really cool thing about this that was innovative is that we're not just looking at whether a farmer adopted or did not adopt a practice. All those conservation practice research papers that I showed you all use a binary variable, whether the farmer adopted or didn't adopt. But that's not entirely how human behavior works. So we created a third category. So did the farmer use the practice? Did they use the practice and have no plans to use? Or did they use the practice and then, or didn't use the practice, but they're open to future use? So that second category there is really looking at openness or intention. So we're recognizing that farmers don't think in the binary. There's a stage there where they are initially open to the use of a practice, and that's important to measure. So we got really good response rates on the 5 different years of the survey. And importantly, 3,200 respondents completed the survey at least twice, which allowed for an assessment of no-till and cover crop adoption over 2 to 3 year periods. And so here's what I really want to hone in on with the time that I have left. This is looking at cover crops. So what we see is a lot of movement between categories. So for instance, if you look at, so year 1 is on the left, year 2 is on the right, and we show where farmers go from year 1 compared to year 2. So for instance, 15% of farmers that adopted cover crops in year 1 no longer are using them in year 2. They're open to adoption, so they might use them again, but they're not using them. And almost 5% went from adopting cover crops to not adopting them with no plans for future use. What happened to those people? But on the other hand, some of the non-adopters moved into well, maybe now I'm actually open to using cover crops. And some of them went from, I have no plans to use it, to the next year, they're like, you know what, I'm going to go ahead and do this. And then the open to adoption went both directions. So there's a churning effect here, that what we're learning is that conservation adoption is not static. It's absolutely changing over time. Same type of pattern with no-till. We're seeing more adoption continuing over time, but certainly you're still getting this churning effect. So again, in year 1, almost 8% of farmers stopped using no-till in the second year, and almost 5% decided that they not only were not going to use it, but they had no plans to use it in the future. So what does that do over time? Basically, it means that if farmers had continued using these practices of cover crops and no-till, and if farmers in general adopted them at the same rates, our rates of adoption would be much higher. But instead, what we're having is this churning effect where farmers are using a practice, then deciding, well, I don't want to do it right now, I might use it in the future, or even taking another step back and saying, I don't have any plans to use this again. So there's a really important effect. That's having a decrease on the rate of adoption of these conservation practices. So I thought, why the heck is that happening? And I think we need to understand that. So I applied for a grant through INRC, and it was approved to do this project, Incentivizing Conservation, Understanding Opportunities and Barriers that Influence Iowa Farmers' Decisions to Adopt Nutrient Management Conservation Practices Over Time. Academics love really long titles, right?
You got to compare it on the review for the long time. No,
doggone it. What would have been even better if I had a nice long quote at the beginning of that too, and then the long title. Sociologists love to do that. So in this ongoing project, actually, we just started. So we're asking what factors influence farmers to continue using cover crops over time, and what factors influence farmers to discontinue using cover crops over time. I would love to follow this up with a study focusing on no-till, but in terms of time and expense, we just wanted to focus on cover crops for this project. So this is using a different data set. It's the Iowa Farm and Rural Life Poll, which is a survey that has been in effect since 1982. So we're interviewing the same farmers year after year with certain changes over time, of course. But we're specifically looking at cover crop use. And whereas we had the 3 categories before, now we have 4 categories. So we have farmers that used cover crops in year 1 and continued using them in year 2. We had those that used in year 1 and stopped using, but intend to use within the next 3 years. And so that's not just openness, that's actually setting an intention and a plan to use cover crops. So that's getting a little bit closer to adoption. Then we have those that stopped using but are open to future use, and those that stopped using and they have no plans to use. And so I'll be interviewing approximately 40 farmers, so about 10 in each of those 4 categories of practice use or discontinuance. And what I'm asking is basic demographics like, what kind of farm size do you have? What do you plant? Have you done other conservation practices? What % of your ground is leased versus owned? How long have you been farming? Things like that. Ask them about sources of support to adopt cover crops. Did they work with the INRCS through financial assistance or technical assistance? Did they work with extension? Did they go on YouTube and try to learn from other farmers? Things like that. What were the barriers to continued cover crop use? And for those that continued using them, how did they overcome those barriers? And for those that stopped using cover crops, what resources might they have wanted to potentially continue using cover crops? And I also want to understand from these farmers how agricultural and conservation organizations can better support farmers in their use of cover crops. All of the research that I do, I try to make applied so that we can make recommendations to those who are working with farmers in the field as well as policymakers to hopefully increase conservation practice adoption as a whole to address these environmental concerns that we have. That's all I've got for now, and I'd be happy to take any questions. Yes.
Is the use of cover crops tied to any certain type of tillage system, or when you say somebody's using cover crops, they could be doing reduced tillage, heavy tillage, or no?
Yep. Question was, is the use of cover crops tied to any tillage system? And no, we're looking at those 2 things independently, but they are certainly connected and related. And that's something I'm hoping to bring out in the interview project. Yeah, good point. Yeah. Any other questions? Yes.
So, I think you touched on this, but in the the work that's been done in this area, nobody's really looked at over time. I mean, because this is we implement these programs, say, okay, we're implementing this, but then, yeah, I've always wondered, like, okay, do those people continue to adopt? And I think what you're saying is some of them continue, but a fair number of them are kind of not continuing.
That's exactly right. And so I certainly won't make the case that we are the first people to look at conservation adoption over time, but there are very few studies out there and even fewer studies in the Midwest looking at these particular practices. So that's what's innovative about this. And yeah, and to your point, like when I worked for the NRCS, I would do equip programs with farmers to help them plant their cover crops. After that 3rd year was over, we signed off on the contract and we're done. I'm busy working with other farmers. There was no time to go back and do outreach with that farmer and say, hey, are you still doing this? Is there any way that I can help you? So I'm hoping to tease some of that information out of farmers with the interview data to see if that's something we can push for, doing more review and continued outreach after working with farmers. I think that's a big piece that we're missing. Yeah, so you had a question.
Yeah, it was really interesting to see how the perception of practices has changed.
Yeah.
Where you showed like, oh, you're more progressive if you start using pesticides, whereas with the rise of organic farming,
right,
it's almost been flipped.
Yeah.
Have you noticed any other shifts in perceptions through your research?
That's interesting. I think so. You're absolutely right. Perceptions have changed over time. There's definitely still a push to be as productive as possible. So I'm definitely not saying that all fertilizer is bad, all herbicides are bad, that just growing 2 crops is bad. I think in certain situations we can improve all of those metrics that will help the environment. but farmers are really good at producing, corn and soybeans, right? So there's still that push. But what has changed is more of a sustainability concept long term, not just looking at the sustainability of their yields, but will I have enough soil left on my farm to pass this to the next generation, right? Are my kids going to be able to inherit the farm and have a farm to work? I'm seeing more focus on wildlife. So a lot of farmers, hunt, and there's always been an interest in wildlife, but specifically doing like hedgerow planting or pollinator planting, or even, potentially a benefit of cover crops is that it can be some wildlife feed and that sort of thing. So more of a focus on wildlife. And there's more of a focus that I'm seeing in certain farmers of downstream effects. So a lot of these practices that we're asking farmers to implement really don't benefit their farm. They have a benefit for people downstream, but there's more of this realizing that I'm not just here on my own, I'm within a watershed, and that I have a duty to, protect the resources of people downstream as well. So we're kind of seeing more of a movement that way. Yeah, good question. Was there a question in the back?
I enjoyed your presentation.
Thank you.
In fact, in the second project, the type of questions you had developed, they are really good questions.
Thank you.
My follow-up is, are you planning to write third project to look at some of the lessons you might have learned from a set of questions to propose a policy to the federal government or to the state of what it could do in the adoption of these practices?
First of all, I appreciate that. And I think that's a great question. In all of the research papers that I write, I always try to have some recommendations for policy. I think I probably could, with the accumulation of data that we're getting from lots of different studies, probably just write a policy paper about all the lessons that we've learned. I also try to get outside of the silo of the university and work with external partners. And so I'm doing a project with the Soil and Water Conservation Society, which is a big advocate for farmers and for conservation professionals. The cool thing about people like that is they have connections in Washington, D.C. that I don't necessarily have. And so they can actually bring our research to the people that are making those types of policy decisions. So that's definitely something I want to focus on in the future. Yeah, I don't want to hog all of the question time. Is there any questions for Grace or anything online?
There is a question online. So do you get the sense that any of the farmers interviewed also connect the issues of water quality to their local environment or quality of life, for example, drinking water?
Yeah, that's a great question. So absolutely, I have talked to farmers that are making the connection that what they do on their land doesn't just affect the crops that they're growing, but it affects the greater watershed. I kind of mentioned that, but in terms of like safety, I've had farmers realize that there's an impact on their groundwater. A lot of farmers in rural areas are on wells, and they're seeing the studies about nitrate being in the water and bacteria being in the water. They get the reports that half of Iowa's surface waters are affected by either algae or sediment or nutrients, and there's swimming restrictions and things like that. So I think there is more of a connection to the farming practices and water quality, and that's not just a downstream problem. That's something that affects all of us, including the very water that they're drinking on their own farms. Yeah, that's right. Anything else from online or anything for Grace? Yeah.
I think an interesting combo you can do is if you're doing wetland, you're wanting to look at like how to motivate farmers to get more wetland. Maybe the hunting part would definitely, because water, I mean, if I'm thinking of hunting deer, first thing I'm gonna look up how many ponds you got.
You bet.
Because if there's pond, they're probably gonna go drink it.
Right.
So, and I mean, I know it's what is it called? I don't look the word for it. The water that you're wetland.
Oh,
wetland. I'd call it a pond. Like,
Yeah.
so,
Yeah.
So like if you're telling a farmer like, hey, if you want to go deer hunting, well here's now you can put a pond on your land. You're helping save the environment, and you're also like you can just be lazy and put up a stand and watch the pond. So,
yeah, I think I see a future research collaboration is what I'm hearing there. Yeah, let's get together and talk about wetlands. Yeah, awesome. Yeah, really good point. Finding ways to incentivize farmers to do these practices that are not just going to benefit society and natural resources in general, but that are going to benefit them, that are going to work for them. Some of you may know about the prairie strips practice that was developed here at Iowa State through Lisa Schulte-Moore's lab. That's now been adopted at the national level as a way to reduce soil erosion, improve soil health, improve water quality. And also there's tons of wildlife and pollinator benefits for those prairie strips going through your crop field. And my colleague, Derek Franklin, is doing research, interviewing farmers about their perceptions of having these practices on their land. And so they're saying things like, I love just walking out in the prairie strip. It's beautiful. I love seeing birds out there. One farmer said, I picked flowers from my prairie strip and used them in my daughter's wedding. So like we have to figure out whatever it is that incentivizes farmers and push that for sure. Yeah. Yeah.
Got one more, Chris, maybe
Okay.
online.
Okay.
Somebody that's on the social science side. Do you think the start-stop adoption cycle in your data has implications for establishing more sustainable norms among farmers?
Oh, that's interesting. Yeah, so we do know that in sociology, norms have a huge impact on what farmers do. The coffee shop effect is something that we talk about a lot. Farmers get together and say, what are you doing? What are you doing? That can have a positive effect on conservation adoption as farmers try to keep up with each other. There's also a negative effect if you're doing something different that nobody's ever done before. They kind of look at you and they're like, what are you doing? Why does your field look so weedy? Those aren't weeds, that's a cover crop. That sort of thing is definitely an effect. So I think you hypothetically could get more continued use if farmers in a community were doing that. And there's probably some regional effects, something that we would like to dive into in the second stage of that first project that I talked about. We know there's county level effects where you have people advocating for that and really building a community of sustainability for sure. There was something else I was going to say about that. I apologize. I'm not thinking. Oh, the other thing I wanted to mention is that there's also a thing here that sometimes farmers just they will skip cover crops for a year and even no-till because of the weather. So that's something I want to tease out in my interview data. Some of these farmers have tried it, didn't like it, and they're abandoning it, right? But some of them might just be a practical management thing that they're pausing it and they'll continue it again under different conditions. So that's another thing I wanted to.
Okay, so just for time, I think we're going to stop with questions there. Go to our two advertisement slides. All of our seminars are recorded and can be found on the INRC website. We started our seminar series in 2020. All of those can be found up there. And then our next seminar is March 12th at 3:10. We have two PhD students, one talking about cover crops and the other who is going to be talking about microbial communities in wood chip bioreactors and corn cob bioreactors. Right, and that is it.
Thanks, everyone.